Hey everyone, just to take off from where we left off last class, I'm going to get into protein structure here. So we left off just finishing up here in terms of understanding that to create a protein, we have to join amino acids together. So here we're joining two amino acids through a dehydration synthesis, right? And again, that means the removal of water to join two monomers together, and the linkage between those two monomers is called a peptide bond, right? So it is a covalent bond, so that's really important, right? Covalent bond, remember carbon and nitrogen are both nonmetals. So as we move to protein structure, we're going to look at how proteins take shape as amino acids join together. All those things we talked about in terms of polar, non-polar, hydrogen bonding, okay? Um, all of these different types of bonds, all of these different types of forces of attraction or repulsion are going to play a role in how this protein takes shape. All right. So there's four levels of structure to a protein, and they are named accordingly. So primary being the first, secondary, second, tertiary, third level, and quaternary is the fourth level of structure to a protein. All right. So the primary structure refers to the sequence of individual amino acids characterized by individual covalent peptide bonds. Okay, and so in the picture off to the right here is, we would refer to this as the primary structure, this diagram right here. And although it might be hard to see, they've just abbreviated the names of some different amino acids, valine, it's hard to tell there, maybe aspargine, isoleucine, serine. So they just abbreviated the names, but trying to show that they are joined, each amino acid, by the covalent peptide bond. All right, and that's what we refer to as the primary structure. Sometimes you'll see it just written as a 1 with a little degree sign. So when we move on to secondary structure, and you see it with a 2 with a little degree sign. Secondary structure is this picture over here, secondary, and also this picture here. There's two different shapes to the secondary structure. So one is the what we call the alpha helix. So that's the first one. Secondary structure is characterized by a helix, which is what we call a spiral shape. So it's characterized by a spiral shape or what we call a beta pleated sheet. Okay, and so I should say that this helix is characterized by what we call a alpha helix. So just the Greek letters of the alphabet, alpha and beta, one being the, A, the first version, B, the second version of the secondary structure. So it's characterized by these alpha helix. So here's this spiral here, and over here is this sort of beta pleated sheet. And what determines that is, so it's caused by hydrogen bonding in adjacent amino acids. So remember, in those functional groups, the carboxylic acid and the amino group, what we need to remember is that some of these are going to have slightly positive ends, slightly negative ends, and as you join them together, these attractions will twist them either into this spiral or into this beta pleated sheet, okay? Third level of structure is what we call tertiary, so characterized by 3 and a degree. And so what's going on here now is kind of the creation of this three-dimensional shape starting to take fold, pardon the pun. So down here we're into the third level, tertiary, all right? A unique three-dimensional shape, 3D shape, caused by a variety of interactions between R groups of individual amino acids. All right, and so individual amino acids. And so these interactions, they could be hydrogen bonds. They could be a particular type of bond called a disulfide bridge, which is a sulfur in one R group of an amino acid may strongly attract a sulfur in another R group and form a very strong, this is actually a covalent bond. All right, so disulfide bridge or other ionic and covalent bonds. But they exist between interactions of the R groups as opposed to in secondary structure, hydrogen bonding between adjacent amino acids, between those functional groups, the amines and the carboxylic acids. Then finally, it's not shown in this picture over here, but the fourth level of structure is when a protein has two or more tertiary levels of structure that interact. Okay, so quaternary is the interaction of two or more tertiary levels of protein structure in one molecule. Okay. So I just paused the video momentarily there just to flash back to the very first page of our notes in this chapter, and we discussed this protein here, which is hemoglobin. And what I want you to, I'm pointing out here, the quaternary level of structure, each of these different colors, you got a purple here, a green here, blue over here, and red up here. Each one of these individually is a tertiary, but collectively you have four different tertiary level proteins all interacting collectively to give this quaternary level of structure in the protein hemoglobin. Okay, so this is one protein molecule, but it consists of four what we call, so there are four subunits, and each of those subunits interacting in a way to make one functional quaternary level of a protein. At this point, I would encourage you to watch this video. In order to watch this video, you can go to the book of videos on our Moodle page under the under chapter 3. So if you go to the videos here, you will see that particular video at page 5 on protein structure. So I'd pause the video at this point and have a watch. Okay, hopefully that video was helpful to give you kind of the visual of how these four levels of structure come together to determine the overall shape of a protein. Now, what's important here, like the main theme I've tried to emphasize here, besides homeostasis, the theme of human anatomy and physiology, in order to maintain homeostasis, I've discussed how important structure serves function. And so again, here in terms of this overall protein shape, whatever unfolds, so to speak, determines a very unique shape to every single protein. And this unique shape dictates its function. If a protein loses its shape, we call this denaturation. Or we say, you know, the protein has denatured. All right, we've covered some basic chemistry out of chapter 2 and talked about some various types of chemicals that could impact the environment of our cells and the fluid around our cells. So I'm going to pause the video for a second. I just want you to think of what could cause a protein to lose its shape. Okay, so I'll pause the video, give that some thought, and then start the video to find out. Well, if you've thought of things like changes in pH, all right, so the addition of acids or bases could have a strong impact on the shape of the protein because it could disrupt hydrogen bonding or other types of interactions that we brought up above here that create this three-dimensional shape. So changes in pH for sure, changes in temperature. When we tend to heat things up, right? Heat can cause changes in those interactions. And again, changing those interactions, hydrogen bonds or disulfide bridges, etc., will cause the protein to lose shape. Other things like high salt concentrations, okay, because again, when we think of salts and we put salts into solution, remember that something like sodium chloride would dissociate into sodium and chloride ions. And again, these charged particles could influence the shape of that protein. And lastly, heavy metals, okay? So heavy metals are compounds such as maybe copper or mercury or lead, okay? These metals tend to have high charges. And again, same reason. These charged particles can influence the hydrogen bonding and those other weak interactions that form the shape of the protein. And so you can think of, maybe you've heard, another one, cadmium. These heavy metals, they can be toxic in our environment, get into our drinking water, and they can certainly influence the function of our proteins because they alter their shape. All right. So we've emphasized the structure of proteins, how that structure is created and how that structure can be altered. So ultimately, what are the functions of our proteins? What do proteins serve throughout the body? We've talked about carbohydrates and the role that they play in terms of a source of energy for our cells and for us. We've talked about lipids and fats and how they're vital for protection and insulating our organs, how they form the structure of our cell membranes, how they play a role as chemical messengers, as hormones. So what do proteins do? Well, proteins have a rather large list of very important functions. Probably the most important, well, it's hard to say any one is more important than the other, but one of the big ones is they function as enzymes. And so enzymes serve as what we call metabolic catalysts. This means is they help speed up chemical reactions in our body. All right, so enzymes speed up chemical reactions in our body. And every enzyme that exists in your body, enzymes in your saliva, enzymes in your digestive tract, you have enzymes in your bloodstream, in your liver, you have enzymes throughout the tissues of your body, and they're all playing a role in speeding up chemical reactions. But proteins have other roles as well. We have Structural proteins, which provide associations between body parts. So we have structural proteins. Some of these can be within our muscles and what connect our muscles to other components like tendons and ligaments and bones. So we have structural proteins. We have contractile proteins found within our muscles themselves to help our muscles move. We have defensive proteins, which include a whole list of antibodies that our body makes from our immune system. We have what are called signal proteins. Apologies, I had a huge sneeze coming up. I had to pause the video. We have signal proteins, which are best exemplified by something like insulin. And so we commonly know these as hormones. It's signal chemical reactions to take place, right? We also have receptor proteins, which we've seen through cell transport, or we will see coming up. Receptor proteins that transmit signals into cells. These receptor proteins sit on the surface of our cell membranes. We have transport proteins. A good example of that would be hemoglobin, as it transports oxygen throughout our body. But we have lots of other kinds of transport proteins. We have transport proteins that help move fats through our body, because remember, fats are non-polar and not water soluble, so they are sometimes better transported in different means. And we have storage proteins that serve as a source of amino acids for developing embryos. Other storage proteins, an example like ferritin, which stores small ferrets in your body. Okay, just kidding. Stores iron, which is the Latin name for ferritin, ferrous, the Latin name for iron. So that concludes our notes on proteins. I'll pause the video and you can scroll to the next page to pick up on the nucleic acids.